Good day, everyone. My name is Caleb Lewis, and today I'll be presenting on some of the work that I've been doing at the Cocoa Research Center here, relating to the genetic variation of cadmium bioaccumulation. Today, I'd like to focus mainly on the variation of cadmium in cocoa through seasonal and developmental stages. So throughout my time here at CRC, I've published a couple papers. The first one that I published, well, we as a team published, was the genetic variation in bioaccumulation and partitioning of cadmium in cacao. This was the biggest, to date, the biggest study done relating to the analysis of genetic variation of cadmium bioaccumulation in cocoa. So in this study, in this paper, we have shown that bean cadmium concentrations vary between varieties, and also leaf cadmium concentrations as well. In addition, recently, this paper was I only got the official document Friday. We only got it on Friday. We recently published a paper, plant metal concentrations in Theobroma cacao was affected by soil metal availability in different soil types. In this study, one of the things that we looked at in these different soils is what are the main factors that affect plant metal concentrations in cacao. So for instance, in cadmium, we found that soil available cadmium, nickel, zinc, and soil pH affect plant cadmium concentrations in cacao. We also looked at we also looked at plant manganese, nickel, lead, and zinc. We also, for this paper, we also looked at plant iron and chromium, but we weren't able to develop any significant models relating to the soil conditions and relating the soil conditions to the plant conditions in cacao. So moving forward, we have done more genetic work, and we have increased recently we have increased the number of accession screens. So in the first paper, you only screened 100 varieties. In this work that is, well, currently ongoing in terms of the analysis, the collection is already done. 500 plus accessions were sampled and analyzed for leaf and bean metals at ICGT. This was a project in collaboration with CRC and the Future Food Beacon of Excellence. Leaves were collected for genomic analysis, and bioinformatics and SNP identification is going on currently. So in this study though, what we have identified is we've shown a similar range of cadmium concentrations in cocoa at the gene bank. So cadmium concentrations varied from 1 milligram per kilogram to 7 milligrams per kilogram. Some accessions were higher, but the majority of the accessions that we sampled, 500 plus accessions, varied between 1 to 7 milligrams per kilogram in leaf cadmium concentrations. So we also looked at bean cadmium concentrations, and what we found is that most of the varieties varied between 1 to 4 milligrams per kilogram. And this study, of course, as well, it shows that there's variation in cadmium accumulation between varieties. We also correlated cadmium with other metals that we also analyzed in this study, and we found that leaf cadmium was correlated to cobalt, nickel, manganese, and zinc, whereas bean cadmium was best correlated with bean nickel. So these findings represent a repeat of the initial gene bank screening. So in the initial gene bank screening, we found that leaf cadmium was correlated to nickel, manganese, and zinc. We did not find the leaf cadmium correlation with leaf cobalt, but that was because we did not analyze cobalt in the initial study. Bean cadmium was also correlated with bean nickel in the previous study. These metals are nickel, cobalt, manganese, and zinc are essential metals. So the plant has pathways that facilitate the uptake and transport into the leaves and beans of the plant. So the correlation of cadmium with these metals suggests that the pathways that facilitate the uptake of cobalt, nickel, manganese, and zinc may also facilitate the uptake of cadmium in cacao. Of course, further analysis is ongoing. We have additional data, not only the bioinformatics and SNP work, but there's also additional information we collected from each tree that we are analyzing and looking to refine the results. During this time, the work that I've done, I've identified some gaps in terms of how we analyze and how we view the cadmium accumulation problem in cacao. One of the problems was the inconsistency of leaf sampling for metal determination in cacao found in the papers that publish cadmium studies in cacao, in the sense that there's no defined sampling method for leaf samples. So for instance, the papers that I publish, the work that I do, I try to focus on sampling the interflush 2 phase of development. That's the phase that I work with consistently. Whereas I've seen papers that are published and they collect the 10th leaf from the top of from the apex of the flush, and other different mechanisms of sampling leaves. So what we wanted to look at here at CRC is how that inconsistency of leaf sampling can affect the determined cadmium concentration in leaves. There's also limited understanding of seasonal variation in cacao metal accumulation, because cacao is a perennial crop in the sense. So it's a cash crop, yes, but that crop is harvested. The pods from that crop is harvested over 30 years, but the plant is there growing and developing in one plot over a 30-year period. Now, most of the studies that have been done on crops and cadmium accumulation in these crops are seasonal crops, so rice, you have wheat, etc. That's where the majority of the findings, of the major findings that come from these crops. So there's limited understanding of how the seasonal variation that cacao would experience by being in one plot for 30+ years, how that will affect the metal accumulation. The third gap is fruit sampling focuses on only a subset of the cacao fruit. It's focused on the economical part, which is the cotyledons. Now, while the cotyledons may be the economical part, and it's understandable why we would focus our effort on trying to reduce the cadmium concentrations in the bean itself, as the bean develops, it develops as a pod, not just the beans themselves. So the pod husk and the testa and the cotyledon as well, they develop throughout that fruit development period. So what I wanted to look at is looking at cadmium accumulation in the entire fruit itself. And that leads us to the objectives. First objective is to investigate the effect of leaf development on cadmium accumulation in cacao. Determine if cadmium accumulation in cacao varies with time, and investigate the partitioning of cadmium between the pod tissues of cacao. So for the first study, this is the leaf development study. I used 5 high cadmium accumulating varieties, CRU72, ELP1S4, GU335P, PA293, and TRD94. Now, the 3 accessions highlighted in red, CRU72, GU335P, and TRD94, I'll be focusing on those 3 for this part of the presentation, because due to COVID-19 and the restrictions, we weren't able to get all of the analysis done in time. So I'll be focusing on the 3 that I have the data for. 3 trees were selected from each accession. And from each tree, 5 to 10 leaves were collected from each of the following flush phases: interflush 1, interflush 2, and interflush 3. Interflush 3 collection was split into 4 groups. Group 1, youngest leaves on the branch, who had the interflush 3 phase. Group 2, youngest leaves on the branch were at the flush phase 1. And group 3, youngest leaves on the branch were at flush phase 2. Whereas group 4, youngest leaves on the branch were at interflush 1. So a total of 6 leaf samples were collected per tree. In the following slides, I will explain what the breakdown of the interflush tree collection. So first I'd like to show you the interflush 2. So based on the paper by Greathouse et al., where they identified the flushes based on the stem itself. The reason why they did that is because the leaves may look mature based on not only their developmental stage, but also the amount of sunlight they're exposed to. So based on their location in the tree. So a leaf sample that is more exposed to sunlight will start to look more mature than the developmental stage it is at. But that does not mean it is at that developmental stage. So what they focused on looking at the stem. So at the interflush 2 stage, the stem, the top of the stem, at the side that's facing the sun, that's more hardened, more mature, whereas the back side of the stem itself is still green and shows, of course, some signs of immaturity. So this is the interflush 2 phase of development. So the interflush 3 phase, as you can see on both sides, so the leaves look more mature as well, but on both sides of the stem, there's that maturity. There are no chlorophyll pigmentations observed on the back side of the stem itself. Interflush, I didn't show a picture of interflush 1, but interflush 1 is the stage where the youngest fully expanded leaf stage. So that stage is, I'll go back to the interflush 2. It's less mature than the interflush 2 stage. Both the top and the bottom of the stem would be green, and the leaves as well are usually softer than the interflush 2 and interflush 3 phases. So we're moving on now to the interflush 3. So these are the interflush 3 at the flush phase 2. So the flush phase 1 and flush phase 2 are the stages where you get most growth of the leaves. So flush phase 1 is when the apex starts to divide and you get new young leaves coming out. Once those leaves are open, they start the flush phase 2 part of the cycle. The flush phase 2 part of the cycle is the longest part of the cycle, and it goes from the small open leaves to the youngest fully expanded leaf stage, which is the interflush 1 stage. So at first, of course, is the flush phase 1, then flush phase 2, and then interflush 1. After interflush 1, then there's interflush 2 and interflush 3. So I sampled interflush 1 and interflush 2. It wasn't possible for me to sample flush phase 1 and flush phase 2 because those leaves, of course, would be too small to get a good representative sample that I can analyze that could have been analyzed. So I sampled interflush 1, interflush 2, and interflush 3, but I also looked at leaves at the interflush 3 stage, but were a bit older than the interflush 3 stage where they have just matured. So the interflush 3 stage at flush phase 1 would be more mature than the interflush 3 stage, and the interflush 3 stage at flush phase 2 would be more mature than the previous 2 stages. So for the method, the leaf samples were cleaned and dried. Dried leaves were then weighed and ground to a powder. Ground samples were digested using nitric acid and analyzed for cadmium, manganese, nickel, and zinc. Chemical analysis was performed using atomic absorption spectroscopy at the Department of Chemistry in the University of the West Indies, St. Augustine campus. So one of the things I looked at was leaf development by mass. So as you can see on the bottom of the graph, I1 is interflush 1, I2 is interflush 2, I3 is interflush 3. F1, I3 is leaves collected at the interflush 3 stage, but the youngest leaves were at the flush phase 1, as shown in the previous picture. And well, F2, I3 is flush phase 2, interflush 3, and I1, I3, interflush 1, interflush 3. What we found is that as the age of the leaf increases, you have an increase in the mass of the leaf. So the leaf gets bigger as the as it develops, which is of course expected. But how does this? The question is, how does this affect the cadmium concentration and the metal ion concentration that is found in the leaves themselves? So when we look at flush phase versus cadmium per leaf, it follows the same trend. As the leaf gets bigger, you have more cadmium in the leaf itself. So in this graph here is the same thing. We look at the different flush phases, but I'm looking at the amount of cadmium in each leaf. So as you can see, the cadmium concentration increases, content, not concentration, increases as the leaf gets bigger. On the graph, different letters represent significantly different cadmium content. Now, what I also looked at, of course, is the concentration of cadmium in the leaves. Now, even though what we saw before, where as the mass of the leaf increases, the content of cadmium in the leaf increases, we're not seeing that same trend here. So what we're seeing is that the concentration steadily increases until the beginning of interflush 3. Even though at that point, the leaf still increases in size, the concentration stays the same. So there seems to be some sort of steady state here where the cadmium concentration reaches its optimal level in the leaf at the interflush 3 phase. Similarly, I saw this pattern in the GU335 accession. So due to time limits, of course, I can't show all the graphs, but I wanted to show, and I can't show all the metals. However, this here is the leaf nickel concentration. So what I'm trying to show is that across metals, we're seeing the same trend, where as the leaf increases in age from interflush 1, there's a steady increase in metal concentration. But when the leaf reaches the interflush 3 phase, the concentration plateaus, resulting in a maximal concentration of metals in the leaves. Now, the conclusion from this study is that leaf metal concentration increases throughout flush phase development and plateaus at interflush phase 3, as I stated before. The leaf mass continues to increase throughout interflush phase 3, but metal concentration remains constant. So there's a plateau of the concentration of metals within the leaves. When sampling, so what this means is that when sampling cacao leaves for metal analysis, leaf development state should be noted and reported. And it's important this is important because as Greta and as also I saw throughout my sample analysis, sometimes we think that the leaf is a mature leaf, but it's not a mature leaf, but it's based on the position of the leaf relative to the sun. So if it's exposed, as I said previously, to more sunlight, it can look more mature. But also, if it is that you're sampling for cadmium analysis and you're not paying attention to the phase of development the plant is going through, the shoot is going through, the leaves that you are selecting are at, then what can happen is that you can collect from multiple flush phases, which can of course cause a problem when you try to determine the amount of cadmium in the plant, in the sense that you would not be actually sampling or determining the concentration of cadmium at a flush phase, but you'll be determining a mixed concentration which could of course affect your reproducibility. So you can now, that can lead to different concentrations being determined when you sample at different periods, simply because you have sampled, you have a mixed flush phase collection for your leaf samples, right? Now, at this point, I can't see what is the best flush phase based on the data that I have to sample, because that relates best to the plant itself. But a study reported by Snoek in 1987, they stated that leaf sampling at the interflush 2 stage is best to, it represents the metal content of the entire plant. It's the best representative sample you could collect to represent the metal content concentration of the entire plant. So I would advise to continue sampling at the interflush 2 stage of development, even though the concentration plateaus at the interflush 3, because the plant is always in a steady state of new to old flushes. Sampling at the maximal concentration point may not be the best method going forward for consistency of results, but also for just estimating the plant metal content. The second method I looked at was the seasonal variation study. I sampled 5 high cadmium accumulating varieties were used for the study. The same varieties as before, but this time I'll focus on the ELP1S4 and PA293. As I said, simply for the same reason as before, where it is that we weren't able to complete the analysis in time due to COVID-19 restrictions. From each tree, leaves were collected from the interflush 2 stage of development, and leaf samples were treated as stated in the previous study above. So from the results, what we saw is that there seems to be a pattern. Now, I'm still analyzing the data and collecting the data, but what we can see is that these 2 accessions show a temporal pattern where it is that it seems the cadmium concentration in leaves increases from April to August, and then decreases from August to October, and at some point in between October and December, it starts to increase slightly again. While this temporal pattern is consistent between both, the cadmium concentrations between these time periods were not significantly different. So there may be some temporal effect, but how important this effect is in terms of the plant cadmium concentration is still to be determined. So, as I said, no significant variation in leaf cadmium concentration throughout the year. For these 2 accessions, I still have to do the analysis for the other 3 accessions that I sampled. And selection of leaf tissue, of course, is important for consistency of results. Just to repeat what I said before, if I had a mixed flush phase sample, then I would have likely have seen a lot more variability, leading to the impression of large temporal variation that is not really there. So for the last study that I'll report on today, I looked at pod development. 5 accessions were selected based on length of pod development. These accessions were selected based on the work done by Sergio. She looked at the length of time it takes for a pod to develop from different accessions, and from her study, I selected 5 accessions, one short accession that has a short pod development period, 2 that are medium, and 2 that are long. I selected them based on, of course, pod availability on multiple trees, as well as, of course, the data that she shared with me. 3 trees were selected from each accession. From each tree, 3 to 5 pods were collected at maturity. Pods were separated into podhusk, cotyledons, and tester. Each sample was then cleaned, dried, and weighed. Dried samples were then ground and analyzed for cadmium, manganese, nickel, and zinc. So here I'm just going to present the cadmium data. What I found is that So just to explain this graph, the yellow bar is the pod content. So that is the content of cadmium in the entire pod husk, beans, tester, everything. Whereas the gray bar is the pod husk cadmium content only. The orange bar is the cotyledon cadmium content, and the blue bar, of course, is the tester cadmium content. So as we can see, there is variation between in cadmium distribution between varieties. If you look at IMC 94, there's no the concentration of cadmium in the cotyledons is almost the same as the concentration of cadmium in the pod husk. Whereas PA 289, there's a large difference where the pod husk has a lot more cadmium in it than the cotyledons. So distribution, during the pod development, the pod husk would develop first, and then the beans would develop later on in the pod development cycle. So what is likely is that different varieties have different lengths of these different periods. So one variety may have a longer pod husk development and a shorter bean development, and vice versa. And this may play a role and a major part in the cotyledon cadmium concentration and content, which of course is most important, as this is the main economic part of the plant. So in conclusion, to conclude, cadmium accumulation in cacao is correlated to nickel, manganese, and zinc concentrations in the plant. That's from the first published study. Stage of the flush phase can affect the determined cadmium concentration in the leaf. Sampling leaves at the interflush 2 phase of development is still recommended. And it's also recommended that people note and report the phase that they have sampled. Temporal variation in cadmium accumulation in cacao can affect the terminal leaf concentrations. And both cadmium concentration, bean cadmium concentration is likely influenced by variations in length of pod development. Thank you.